Hello everyone, it's Dr. Ali again for plant structure and function. We're going to continue with leaf and seed. Let's have a look at the anatomy of a leaf. First off, the first layer will have the cuticle, which acts as a permeability barrier for water and prevents the contamination of plant tissues with outer materials such as dirt or microbes. And then we have the upper epidermis. It's a single layer of cells that permit most of the light to pass through the leaf and is covered with the waterproof cuticle there. And then next we have the palisade mesophyll cells or palisade parenchyma cells. These cells are filled with chloroplasts. Usually there's a lot of chloroplasts, and the contents of these cells are the ones that are responsible for photosynthesis because they have a lot of chloroplasts in them. Next we have bundle sheath cells. So bundle sheath is actually they give strength to the vein. Where's the bundle sheath? Bundle sheath. So bundle sheath here. Bundle sheath. So they give strength to the vein, giving it a stiff frame to support soft tissues of the leaf blade. And then, of course, inside the bundle sheath, we have the xylem that supplies the water and minerals to the other cells. So it sends nutrients up. And we have the phloem. So needed to move around the synthesized food. It sends nutrients down. And then next down after that we have the lower epidermis here. So it holds most of the stoma or stomata, which are flanked, the stomata here, which are flanked by guard cells. So another example, guard cells here. And then we have the sponge mesophyll, spongy parenchyma cells. So they are temporary storage structure for sugars and amino acids made in the palisade parenchyma cells. So the spongy mesophyll cells are more spread out than the palisade parenchyma cells because there are intercellular spaces, air spaces. So if there's example here, air spaces that contain fewer chloroplasts than the palisade layer. Let's have a look at the differences between a dicot and monocot leaf. In dicots, the veins would be net-like, whilst monocot parallel. So this is due to the presence of the vascular bundle. Monocots have larger vascular bundles, while dicots have large and also small vascular bundles. In terms of intercellular spaces, you can see dicots have large intercellular spaces, while monocots have smaller intercellular spaces. In terms of stomata, dicots have stomata at the lower surface, mostly the stomata in the lower surface, while monocots have both upper and lower surface of the leaf. Leaf surfaces are dotted with millions of stomata. So stoma is the plural is stomata, and then there's sometimes people call stomate also. So it's a pore found in the epidermis of leaves, stems, and other organs that controls the rate of a gas exchange. So this pore is bordered. By a pair of specialized parenchyma cells known as guard cells that are responsible for regulating the size of the stomata opening. So because control requires movement, and movement of course requires energy, these cells contain numerous mitochondria and chloroplast organelles. So basically in the leaf, they are actually the only cells in the epidermis that is actually green. Finally, let's have a look at the seed. So the seed in a plant is the part that develops from the ovules after fertilization. So they are enclosed in a fruit, typically which develops from fertilized ovary. So the seeds are are formed as a result of sexual reproduction and contain the young embryo which can develop into a new plant. So seeds of different plants may vary in many ways, but the basic anatomy remains the same. So let's have a look at this, the basic anatomy. So a typical seed consists of the following parts. First, we have the testa, which is the outer coat of the seed, which protects the embryonic plant. And then we have a micropyle, which is not labeled here, is a tiny pore in the testa that lies on the opposite tip of the radicle. It permits water to enter the embryo before active germination. Next, we have cotyledon. So in some plants, this contains high quantities of starch and provide the source of food for the developing embryo prior to germination. So in other plants, this is performed by the endosperm. So in the monocots, there is only just one cotyledon, while in dicots there is two cotyledons, depending on the type of germination. So the cotyledons remain below underground or pulled up above the ground. Next, we have the epicotyl. So the epicotyl is the tip of the cotyledon which forms the shoot tip of a growing plant or plumule. And then we have the hypocotyl, which is the other lower tip of the cotyledon, which forms the developing root to a plant or radicle. So radicle is the embryonic root which will develop into the primary root of the plant. It's usually the first part of the embryo to push its way out of the seed during germination. Finally, we have the endosperm. So in many plants, a separate part of storage of starch develops, and this is called the endosperm. So mostly we can see this in maize and wheat or monocots. Okay, so that's all for plant growth and development. So we have looked at stem, leaves, roots, and also the seed. So that's all for today. Thank you. Bye.